Thomas Meisel will be talking about the challenges of nanoplastic analysis. I would say a topic near and dear to our hearts. Please.
So thank you very much, Michelle, for the invitation, and I'm very happy to be here. So welcome the online participants. So lots of has been said already. I'm also here as part of priority here, so I'm also interested in establishing connection here to your network here. And I'm here also as an analytical chemist, and I'm the master one who is complaining about data quality. And maybe that's why Verena, she was worried about the data quality, the quality assurance slide. So today, what I noticed is you always talk about micro nanoplastics, but in reality, you're only looking at the microplastic side and very little about the nanoplastic side. And there's good reason for this. And okay. So normally you could start the talk with a joke, but this is not a joke. This is reality. So you know this already that we know that we mostly look at 10 micrometer particles and larger, and the smaller ones are often left out. And the good reason why we want to look at smaller ones. So I won't be you're surely aware of that. And so I will go on to a joke. So there's a classical joke here, and it's an older person here, but it's like the young one here. So he lost some money, and the police asked him if he dropped the money here, and he said no, he dropped it a few blocks down the street. And he says, so why are you looking for it here? He says, because the light is better. This is something we observe very often in science. It's called the street light. And this is actually an observational bias. So we actually look there where it's easier to look and not actually to where we should answer the questions. We look at microplastics, but we know that nanoplastics are the ones we should be looking at. This is a good reason because it's so difficult. Microplastic is very difficult and it's not easy. It's lots of light there, but it's not easy. But nanoplastics, that's really tough. And that's a good reason why we have not many presentations here on nanoplastics. So what I'm interested in here is if you look at the distributions, and this has been touched on already by you, that this is a typical scientific view. So you got a log scale here. So you got here 1 millimeter here, 100 micrometers, 10 micrometers, and you got 100% abundance here. And it looks simple here on the scale here, but in reality, it goes up exponentially. It's a logarithmic scale. But what we see here, we know this hopefully, that the smaller the particles are, the more there are. And if you find 100 particles, this was touched in the first talk by Ian, if you find 100 particles there and you compared it with billions of particles on nanoparticles, this is not fair, this comparison, because if we can assume that the further we go down in size, the more we have, and you're comparing particles with larger particles and with smaller ones, which you cannot do. And my question here is, what happens afterwards? What is below 10 micrometers? We know what's happening above 10 micrometers. And by the way, the ISO standards that are currently developed, and they're currently only at the level of routine analysis at 20 micrometers. This is a very large, actually not what we should be looking for, but this is lots of light there and it's easy to see. But the good news is we know the bad news actually. When there are large particles, we know there must be smaller particles, but we don't know what's happening below 10 micrometers. In this case, even below 1 micrometer. So just to start simply, we looked at baby bottles to find out what's going on in the nanometer range. It's a very simple experiment. And the reason why I want to look at this, it was a hype here on the study of baby bottles. They realized that following the WHO recommendations of preparing your infant formula in baby feed bottles or infant feed bottles, you release millions of particles. And what worried me a bit here is this ups and down here. We could not reproduce, we get smooth patterns here, but this is not question here. But you can see already here, they saw already going down to the micrometer range, that the particle numbers increase already exponentially, but we don't know what's going on below. So this was a reason for us to look at the range smaller than 1 micrometer. Sorry about this. I mean, Ian already touched it for, I guess, for the one looking at PM 2.5 and 100 nanometers. There's a different, there's a question where does a nanoplastic range start? So I use the ISO definition, which is 1 micrometer. The other definition, which is 100 nanometers. So obviously there's a legal problem here, but I'm not touching this. For me, it was below 1 micrometer. So we've been looking at a new technique here to find particles in smaller range, maybe 100 nanometers even. I'm not going into detail here, but just comparing the results from optical counting on filter papers. You can already see the particles released from the bottles go up exponentially, and even in the nanometer range, it goes up exponentially. And we again in the hundreds of millions of particle range. So this is our expectation that going down to the nanometer range, you will go up exponentially into many more particles. The good news here is only if you use glass bottles, the particles are much lower, and silicates are probably much less toxic. I'm not talking about toxicity. I'm an analytical chemist, so it is, I'm not talking about this, but you can count much less particles when you use glass bottles. So there's good news here. But also looking at other products, like female hygiene products. The recent paper here, we had a presentation in our network showing that from all sorts of female hygiene products, you're releasing also millions of particles. This is per milliliter even, so there are billions of particles being released through the use of the contact of plastic with the female body. So so we know now, believe that for simple matrices water, you find billions of particles. But so this spurred a lot of research in nanoplastic range, so it's lagging behind, as you can see here. But we are, this is picking up now, the nanoplastic research. But again, the reason why the study is much more difficult to analyze. But looking what the microplastic field is, it's dominated by environmental sciences, and toxicology is, it doesn't matter, but you can see it in the center, is an important part. But going down to the nanometer range, nanoplastic range, it's toxicology picking up here in importance. So we know it is important to look at nanoplastic. And but there are many problems here. But the reason why they're interesting is they, with the penetrating biological barriers, they're able to absorb significant amount of pollutants, but also bacteria. We know this, and extremely reactive, which will make you worry about how do they react in non-simple matrices like human tissues or in a wastewater treatment plant. We don't know actually, and that they also avoid are not removed properly, and maybe even more released into the sea. So normally we look at microplastic, we know about the microplastic release, so we can assume nanoplastics are also released, and far more difficult to quantify. And so the question is, we saw in a simple matrix water, we can see billions. Does it? Are they even more complex in the environment, in more complex matrices? So we have to understand the process, the formation, the fate, and also to understand the experimental constraints to be able to study this. But so the question is now, does it go up exponentially to the lower nanometer range, or does it drop off? Does it agglomerate and it disappear? Actually, I have no answer for this, but it could be that it doesn't, in the environment, doesn't go up as dramatic as we could expect in water. In seawater, we know it behaves differently. It could happen that it disappears. We actually don't know. So to study this whole field of nanoplastics systematically, and I'm related to this metrological view. I'm the one trying to prepare reference materials, and also the nasty one who is complaining about the data, as I mentioned. But as you know, following this route here, it will years and years will be over until you start starting actually doing exposure experiments here. This is not what you want to actually be faster. And if you look at the NIST webpage, it's from 2019, they didn't update it, but as you can see here, they were planning from 2019 to 2024, just before applying them for risk assessments. It takes them 4 or 5 years until we get there. So if you want to study all these parameters all at the same time and not following this route, you run into many troubles here that we mentioned already, and making it all very complex. So I can skip this. So what I observe in reality is, instead of following this route, you have a method and then straight away apply it to your research your research question without having validated methods. And because you want to have data published. Yeah? And then you realize maybe your method you developed is not the best, and you go back to method development, and then you try more experiments and so on and so on. And so this is a little bit more of a mess, but this is reality. And this is actually how science works. You cannot wait till methods are standardized, harmonized, and the reference materials are available. They will always be behind. So this is, but this is not an excuse to publish whatever you want. Methods, and this should be followed, and also following quality control and so on. So there is other ways. You can always excuse that no reference materials available. But but going back to my question, also Michel asked, with microplastic, I think we're not so pessimistic, but with nanoplastics, I'm fairly pessimistic. We're actually right in the beginning here. And we, the ones interested in nanoplastic research, we are now developing methods to quantify and characterize particles in model systems. And not only counting the particles, but also characterize them chemically, so identify what they are. And for this, we would need particles, make them visible. So to move them out of the street blocks down in the shade into the street light, we would have to make the particles visible. And Verena already talked. One way of doing it is by having radionuclides to label them. But you know, the ones with fluorescence dye, there are problems there. I'm sure you're aware of it. And the other ways of using a core with heavy metal in it, or the metal more easily detected with other instrumentations. But they have to have certain requirements. Verena has already talked about these. And this is something I want to follow. This route is at our university, is we want to create nanoparticles with a core, mainly of silicates, and synthesize plastics on top of them to make them, disguise them as real particles with different polymers, and to make it then tailor-made depending on the instrument you want to use, mass spectrometry or different AFM methods or whatever, to make them visible. This is only the first step. This is far away from any other study looking at real samples, because we know we are not there yet. So this is the reason also why I talk here. So if someone is interested in collaborating here and having ideas what particles you need, then we can think about how to produce them for your experiments. And so that this is the current state. This is quite a bit of mess, and we're just right at the beginning of our in looking at nanoplastics. This is the reason why we haven't got so many publications, and that's why also during this presentation on microplastic I have to look at, because there's plenty of light yet, not enough, but there's plenty of light to see something. And so just to sum up, was able to be shorter here because it's late already. And lots has been said already. Lucas wants to present his talk. And so, the basic message here is we've just started looking into nanoplastics and many unresolved issues, and it's currently actually impossible to properly evaluate any potential risks of nanoplastic. You can do experiments, but it will not be useful for any regulations. And I think I can end here just by thanking my co-authors here. And just for fun, I looked at artificial intelligence. This is something coming up now in the Austrian media. And I just asked, it being in this case, what the future of nanoplastic research is. And this is what is here. And it was quite smart, actually. Okay.
Any questions or comments? So I have a few. I think that it's incredibly challenging. From what I hear at every meeting is that how difficult, how expensive it is to detect, well, not even characterize, but just count. Do you see things moving forward? You know, like, are we looking at a year from now or 5 years from now? Because it's very hard to do our work without being able to enumerate the number of particles in the environment and also in human tissue. Do you have any thoughts about that?
Yeah, so as I mentioned before, that the science moves faster than actually development of reference materials, but we haven't even produced proper reference materials for characterization. But even if we don't know, if we even can see it, maybe it dissolves completely in the tissue. We don't know, maybe we could never see it. But we are not there yet, and it will take years again. Yeah. As you saw on this graph, comparing the nanoplastic research and the microplastic research, there's a lag of 5, 6, 7 years. Yes.
Amazing.
But just I want to try that we should be realistic. It was my basic message.
Yeah.
Not to expect too much.
Yeah, well, we have a project that expects a lot, and the European Commission that expects a lot. Any comments? I don't want to keep everybody. I think maybe afterwards we'll have a chance to talk to some of the people in our group who are facing these challenges. Yeah, that would be really great. So thank you very much again. Last but not least, we have human health. Really nicely addressed by Lucas, and his title is Does MNP Induce Cancer Formation and First in Vivo Data? Lucas is part of our Austria team on nanoparticles or micro nanoparticles. And did you find it?
I just was curious and looked up microplastics and cancer in our PubMed, so this is our browser for many research, and there was basically no publication, and I was really shocked and team, and then I had a broader look, and really there was no publication out about plastic and cancer at all. So I thought, well, why not go back into our big resources we have? Let's say our hospital with 2.5 million cancer samples from the last 100 years ago, with the medical history. So it would be basically, an idea just to start to look at the contaminations throughout the last decades, and if basically plastic contamination would be associated with worse clinical outcome, let's say in cancer, because I'm cancer specialist, I'm working out with these cancer models. And that was basically the start. And then somehow it came to Thomas, Michael, who just talked, and he was the only one who said, yeah, well, we can use paraffin material to detect plastic particles there. This is in principle possible. So this was basically the start of this whole project. And basically, then we put everything together. This was I gave the talk in the oven at home office place, just 2 months before COVID broke out, and then everything was online, and we wrote this project, and we really got a funding for this. So and therefore I'm here. So and just 1 year ago we got the funding, and then we started with our experiment. And well, I just flip through the slides, and you just can ask me anytime. I just put the latest data together. Verena Pichler with us and Verena Koppers, she did the experiment together. So we basically have a group. And well, so how can I? Please. Yeah. Here you go. Okay. So the big question is microplastics and human health. Well, there was a review on that in Science. This was one of the publications they immediately called the first author, Dick Redhack, who is now also a collaboration partner of ours and in the scientific advisory board of Micro One, the name of our project. And because I was so thrilled by this basically outlook, he gave that plastic particles could really be affecting human health. And the background is that, as we know, plastic decays, and our main problem is the secondary microplastic, which is basically resulting from decay of products we use every day, like car tires or plastic bottles, and this accumulates in the environment and comes into the food chain and then ends up in our body. And at the time we wrote the proposal, we knew from the work of basically one of our partners, Professor Schwabe, who found that plastic is in the human feces. So therefore we focused on colon cancer. The reason, because now we know that it goes into the bloodstream, and we could basically focus on any other tumor. So that was the reason we focused in our project on the colon, and then oral exposure, because 90% of the plastics enter the body by oral uptake. So of course, as you know, we find plastic in nature in different organisms. There's different shapes, compositions, sizes, and that makes it very complicated. We know that from a study in Zurich, this was just published from Switzerland, that car tire operation is 97% of the whole contamination outside, and that degradation of packaging material, is and clothes and color on the wall and so on is also a big problem indoors. And we spent, I learned just yesterday by talking Potsdam, that indoors is basically a person spends half an hour outdoors. So we are spending most of our time indoors. So the contamination here, like the carpets and so on, are full of maybe those plastic particles. And we consume basically 5 gram plastics, microplastics every week. So, well, that's where we started, and we no longer focus on the colon itself, on the epithelium, but also on, we know that in the feces we have billions of bacteria, and through the whole sequencing analysis we know that this and the change due to, for instance, chronic inflammation. This is very important for us, and without those bacteria, we couldn't basically live, and we become very sick when we change those microflora in the gut, which is also a topic of our project. So basically, this is the workflow. So we basically want to investigate the plastic particles, starting here, and then we want to look at different processes which modify the plastics, like biofouling, contaminant absorption and droplet transfer, and then investigate if this or when uptaken into the body could lead to cancer formation. And of course, we have so basically the key questions are: does microplastic promote tumor formation? So not only the development, but also the spread, the dissemination, metastasis formation, and aggressiveness of colon cancer. And does this also is associated with microbiome alteration? And how does this work? So also mechanistic questions, of course. And this is our setup. So on the one hand, like Verena already introduced, that we preparation, that we need to do preparation of the microplastics for detection and analysis, like labeling and evaluation. So we have different groups here associated, and we use model systems for colon cancer. We'll come to that in a minute. And then we also want to look at interaction and the human health effects. And there we also like using our big biobank. So we also have the Graz biobank, which is the European biobank with 80 million samples. So we really want to look in a big set of data, which contamination leads to which clinical outcome, and if you can visualize that and associate that with human data. Okay, so just quickly a few data which we have. So when you start a project with mouse experiments, the first thing is you need to write the animal license, and this took some time. So we focused on the license we already had from a colleague of the Cancer Institute, and I'm very grateful for that. Otherwise, we would have lost at least half a year. So we just fed the mice with polystyrene particles with different sizes indicated here. So we have 10 micrometer, 1 micrometer, and 0.25 micrometer sizes with different colors, and we just fed it into normal mice. And then what we saw is basically that this already after and with some time slots, and already after 2 hours oral intake, those plastic particles were in the brain. And with a colleague, with a modeling specialist, and this in Debrecen, Oldo Marolovsky, he basically could modulate. In gray, you see the plastic particles which are accumulated, and if those particles have a cholesterol corona, so if particles enter into the bloodstream, you have different, lipids, proteins in the plasma, and he modeled different, coronas, and this lipid corona, which is very likely because we have a lot of cholesterol in our serum, basically. The blood-brain barrier basically sucks up these particles, and this could be one way how these particles enter into the brain. So the model I just talked about is a chronic pruritus model. So you can induce with a substance called DSS, abbreviation, for just 5 days. You basically induce a chronic pruritus, and we just fed those mice orally by gavage, basically the equivalent of what we in average take up in polystyrene particles. The same particles are just basically introduced to you, and after 10 days of feeding these microplastic particles, so we started this chronic pruritus model. So basically we give 5 days DSS, and after 10 days, basically you sacrifice the mice, and we fed the mice for 10 days with microplastic particles, but only 5 days with DSS. But then you get this is a standard model for chronic pruritus, and you get a full-blown disease after that. And I'm very used to this model because it worked for some time. So what was interesting is, so we have a collaboration with the ETH in Zurich and the DSE in Hamburg. They have big synchrotron, and we got plastic particles with a metal core, palladium core, and there we could do an in and output measurement, and here just show the stool samples. The other organs are not processed as the analysis, but what we found that in this DSS treated mice, 35% of the plastic particles are taken up into the organism, and 65% are basically as treated with the pieces. So we found, and it was also published for humans in last summer from Big Weta, that also in humans you find basically microplastic particles, and we find lots of those particles in the blood, no matter if you treat with DSS or not. So with DSS you get a lot of more particles in the blood, but you also see them, and as we saw, as I showed you in the first experiment, that you find it also when you just feed the mice with micro and nanoparticles, particles, just normal mice, and you find it in different organs. Here just these are H&E slides. I mean, this is my work as a pathologist, just judge basically the extent of the disease, and you have the control basically the animal model, then the normal mice treated with micronanoplastics, and then basically the disease model with micronanoplastics, and the evaluation shows that basically the severity of the disease is increased if you feed the mice for 10 days micronanoplastics. And what we also found is that the inflammation is increased. So we found basically specifically macrophages increased in those mice treated with DSS and micronanoplastic particles compared to the controls. And what was interesting is that we have different there are macrophages, but there are different sorts of macrophages, and we just focused on the different sorts because they are more inflammatory and anti-inflammatory macrophages, and the ones which are more critical are called M2. I mean, this is old nomenclature I learned, so I just tell so name them M2 macrophages because they are pro-inflammatory, and you can stain for Arch1, which is a marker for basically for those macrophages, and there are increased numbers of those macrophages in the micronanoplastic colitis model, and we quantified that. And I have to say we have just 5 mice per group, which is a very low number. Normally you need at least 2 times or 3 times that numbers to get in a statistical significance. But what we see is basically that in this treated mice we have really an increase of this M2 macrophages and no real change in M1 macrophages. And what was interesting is that when we look like on the different microbiome compartments by 16 sRNAs sequencing, we found that we get basically a promotion or proliferation of the Enterococcus and Shigella strains, which are pathogenic strains, and these are specifically, increased in the disease model, which of micronanoplastic particles. And what we are doing at the moment is proteomic analysis. This is done by a co-worker Ulrike Reschen in Köln, in Cologne, who basically does proteomic analysis to focus on the changes in this macrophages and in the animal model. So we did also some in vitro experiments just to find out what happens with macrophage cultures if you give them basically micronanoplastic particles. And what we could show is basically that the size for the macrophages is not so important. So if you give them basically small and large particles, there's no significant difference between the M0, M1, and M2 macrophage types. But what we found is that obviously the quantitative uptake by the macrophages is different compared to the small and big particles. So the M1 macrophages basically take up smaller particles in the 2 macrophages, the larger particles, and that's, I think, also interesting. And well, we are just about because the title was about the colon cancer model, so we have just some preliminary data on that experiment, because the problem was that firstly we just got the animal license a couple of months ago, so we could only start with some experiments, and secondly we just got this particles in the food, so we don't give this by oral lavage. We put these microplastics, nanoplastics in the food, so in the pellets, so that the mice are not stressed by the gavage, which we do every day. And so we started rather late, so with 10 weeks of age, which is basically too late. So we should start at weaning age. So this experiment we are doing at the moment. And what we can see basically now, this is a transgenic mouse model with APC-min. This is the oncogene which is mutated there's a point mutation in the APC gene, which is the first step in the oncogenic pathway in colon cancer, and these mice get colon cancer or get intestinal cancer. Yeah, and what we see is if you look at males and females, 4 out of 5 mice die earlier if you give them like nanoplastic particles. We have no significance yet, but it's going in the direction we hypothesized. But we need more data, of course, and these are just the first things we did. So the next step would be that we use another model as well. So we have this genetic model for colon cancer, but also an inflammatory model for colon cancer, and so Verena Koppert is just doing this experiment in my lab with these 2 models to get more insight into how does this work. Yeah. Okay, so we are focusing on the one hand on the process of cancer formation, on the other hand on the inflammatory processes of the immune system, which is a part of the tumor formations. And we know that a tumor is can be viewed as an organ. So you have basically the tumor cells which grow, but you have a blood vessel who give you the metabolic substances the tumor needs, and the immune system, the nervous system, and so on. So it's a complicated organ which you need to investigate. So and we also focus specifically on the immune system because we know that the plastic particles can influence inflammatory process already on the macrophages and also on the microbiome. So that's what is going on at the moment. So it's a process of investigating on different levels, including imaging, of course, like Verena told you before. And we also do behavioral experiments because we have already those mice, and we know from the literature that it might be that this plastic particles also change the behavior of mice. So we know that this plastic particles go into the brain under some circumstances. So we are also including those kind of experiments. So in summary, we can say basically what we found is that micronanoplastics are quickly uptaken into the blood and enter the blood through the blood-brain barrier in the brain. We can detect basically these particles in almost all organs. It increases intestinal inflammation by specifically promoting M2 macrophage proliferation, and we have increased macrophages in the intestinal tissue already after 10 days after basically feeding those mice with plastic particles. Very quickly, we have a group in Graz, because I'm always asked what can we do, yeah? So we are trying with a group in Graz by Vanessa Stammbaum, she's a clinician, and she does microplastic fasting basically by doing a very clear-cut 1 week fasting. So tell the people systematically, the patients to avoid packaging of plastics, plastic cooking gear, so on. So and basically systematically drawing blood before the fasting experiment, and then and taking the pieces of the patients before the fasting and after the fasting, and try to find out what it does. So we also try to involve the public, by and specifically patients who are worried about all this public awareness about the microplastics in our group. And basically, these are our collaboration partners, and together with Wolfgang Watzak, who is the administrative partner here, who organizes all this, and I'm very grateful that he does this. And these are our collaboration partners worldwide. And with that, I thank you very much for your attention. I hope we can already during my presentation, and I hope we can change the world together by collaborating and by focusing on the different aspects. I learned a lot today. I'm very grateful for that, for being here, and thank you for your attention.
Thank you. Thanks very much. Any questions, comments? Andrea, would you like to come up and ask your question?
Thank you, Lucas. I actually got a preview of your presentation on the slides, but I talked to Martin Buckner a few days ago, and he told me a little bit about the outcome, so it was quite cool. I had one question because related to our results, where we saw that in the cancer cells, especially the liver cells, but I think also in the CAR-T2s, we saw the increase in the glycolytic capacity, so basically changing the bioenergetic profile from the oxidative phosphorylation to the glycolytic capacity to the glycolytic one in cancer cells in the function of exposure to nanoplastics. Is this something that you would expect? Is this something that makes sense? Or
Excuse me, did I understand correctly? You mean you switch from the oxidative phosphorylation
through the glycolysis, and we see that the glycolytic capacity, so the ability of the cancer cell actually to respond with the acute energy production in times of stress.
May I ask which kind of tumor cells you are talking about? Because they're really different. For instance, prostate cancer or also brain cancer, they start off with oxidative phosphorylation and then switch to the Warburg cycle.
Those are liver cancer cells, HepG2 cells.
Okay, so and this is in vitro data.
Yeah.
I mean, surprising to me, yeah, but it can of course be that you switch the energy production. And normally, very aggressive tumors switch from oxidative phosphorylation to glycolysis. Yeah, and this also happens in prostate cancer. We start off basically with oxidative phosphorylation and then switch to the Warburg cycle, and because it's for them more efficient. Yeah.
We thought as well, but they didn't really. They just became actually more sturdy to any kind of new stresses that you would put on them. So they actually had even more energy. They were able to produce more ATP than without micro nanoplastics.
But the nanoplastics induce the switch.
Yeah.
Okay. Well, it can be. You know, we know from cancer research that you have if you have a certain threshold of mutation, then basically the cells are so aggressive that they don't respond anymore to any additional, because they are already as aggressive as it can be. So I mean, it depends. Yeah? So you could try with different. If you have a model system where you can do, you know, genetic modifications, and you can try out to switch out oxidative phosphorylation or switch on oxidative phosphorylation again. We are doing that. We have one project which is focusing on that, not nanoplastics, but with other, because for us it's also very important to know how this happens. For instance, in prostate cancer. But it could definitely be.
Maybe we could show you the data. And
yeah, sure. Yes, absolutely. Yeah.
And maybe just a small question. In the mice experiment, how do you assure that your control mice has no micro nanoplastics in their diet? Or
this is a good question, because we have no means to, at the moment, to detect them, because we are working together with Silke and with Thomas on methods to detect non-labeled plastic particles in these tissues, because all of our retrospective cohort, it would be essential to know contamination.
Yeah.
And
the background contamination.
The background contamination of water. This is something which is untold, unfortunately.
Yes.
Yeah. Yeah.
Thank you.
Other questions online or in person for Lucas? I think everybody's pretty exhausted, so it might be nice to just relax after the session, and then people could introduce themselves to you and chat about these things. Anything online?
Hello, this is Anna. May I ask a question?
Yes, please go ahead.
And can you hear me? It's okay.
Yes, perfectly. Yeah.
Yes, thank you very much for this beautiful presentation. I was really amazed by the location in the brain of these microplastics just after 2 hours after oral intake. And I was wondering if it is really the presence of microplastic within the brain tissue, or only, microplastic present in the blood, even if the blood compartment is, I know that very well, quite protect with the barrier, the brain-blood barrier. I mean, are you sure that the microplastic detected in the brain is not only, the microplastic within the blood? And can you tell me more about, also the location, because it appears quite disseminated on your illustration that suggests that it is rather microplastic present in the blood. I don't know if I was really clear, but, it's quite
Absolutely
important
clear,
to establish the fact that microplastic can really be detected within the brain cells 2 hours, only 2 hours after oral intake. It's really, amazing.
Yeah, it was also amazing to us. So we saw that in different tissues and also in the brain, that it is really in the cells and not in the blood, but in the blood vessels or in the blood. But you're completely right. We need to do additional stainings to show that the blood vessels basically and the brain cells specifically stain, and also the glial cells, because the brain also has macrophage populations, and we are staining that at the moment to get more, in-depth analysis in which part of the brain, like in which brain cells, which there are different brain cells around. So we need to do more specific analysis of that. You're correct.
And thank you. Just wanted to add something else, because I'm a neurotoxicologist, actually. So I'm an expert of the brain and in nanotoxicology too. So this is very interesting and a very difficult challenge, to deal with. But there is also something very important. You show a simulation very nicely that shows that it is probable that these micro and nanoplastic may cross the membrane, but if it can cross the membrane from the bottom to to down, it's possible that the nanoparticles made of plastic are able also to get out from the brain and go into the bloodstream. So I mean, it's very important to keep in mind that the crossing of the barrier can be in both sense, if you want. So, and usually people don't say that, but it's very important to have it and keep it in mind. Well, this is my opinion, but
Yeah. So maybe like Verena Fischer showed that this particles, at least in vitro, go into the endosomes and the lysosomes. So of course they could also exit the cell again and go back. You're totally correct, but we don't know yet what really happens with the plastic particles, and what we just did this basically one day time lapse with this experiment, but of course it needs to be reproduced and
it will take lots of time. It's complex question. Yes, of course.
But these were nanoplastic particles, so these were this very small, also 250 nanometers or 259 nanometers particles.
Thank you. Thank you very much.
Are you from Bratislava, by the way?
No, from France.
Oh, really? Great.
Yes.
Greetings to France.
Great.
Thank you.
Thank you very much. Any other comments, questions online? offline. Okay. Thank you. Thank you so much. So I think that we're ready to end this meeting. And I'd like to thank everybody for their great talks and for the audience for contributing and participating in this event. And I would say thank you and goodbye.